Hello everyone, myself Dr. Abhimanyu Kumar Jha. I'm Dean Faculty of Life Sciences at Institute of Applied Medicines and Research, Ghaziabad. This is one of the most prominent colleges in biotechnology and microbiology in India. So today I'm going to teach you genetics. We're going to study the basics of genetics. So let's see what is genetics. Now, what is genetics? Genetics is the study of heredity and variations. Now, what is heredity and what is variations? There are two components, as I told you. It is the study of heredity and variations. What is heredity? Heredity means transfer of characters from one generation to another. From one generation to another, the transmission of characters takes place with the help of genes. We know these factors as genes. Earlier, these were called as factors, and this was the term given by the father of genetics, that is Gregor Johann Mendel. So here let's see. Heredity is the study of transmission of characters from one generation to another. Whereas variations means why we are different from each other, why we are different from our parents. In sexual reproduction, the product is called as zygote, and zygote is not 100% similar to any of the parents. Unlike the product of asexual reproduction, the clone, which is just a replica of one of the parents. Zygote is quite a bit similar to both the parents, but it is different also from both the parents. And variations are the reasons why we are different from our siblings also. Variations are the reasons why we are different from each other. Now, these variations arise due to a process called as crossing over. Now, this process takes place in meiosis. I'm sure students, you might be familiar with what is meiosis and what is mitosis. Meiosis consists of two steps. First is meiosis 1 and the second is meiosis 2. So here, as we all know, meiosis 1 as well as meiosis 2 would be consisting of certain steps like, it's very easy to remember them. It's prophase, metaphase, anaphase, and telophase. Now it's very easy to remember them, just like the first alphabet of all of these you can remember like PMAT. It is P for prophase, M for metaphase, A for anaphase, and T for telophase. So, if somebody asks you, when does the crossing over takes place? Crossing over takes place in prophase. Now, since this is meiosis 1 and this is meiosis 2, in meiosis 1, the stages will be called as prophase 1, metaphase 1, anaphase 1, and telophase 1. Whereas in case of meiosis 2, the stages will be called as prophase 2, metaphase 2, anaphase 2, and telophase 2. Now, our question is, where does the crossing over take place? Crossing over takes place in prophase 1. It takes place in prophase 1. And prophase 1 is having several substages, like there are 5 substages of prophase 1. These are leptothene. See, this is something which you study in cell biology or cytology, whereas the heredity part you study in genetics. But since we are going to talk about heredity as well as variations, so automatically crossing over needs to be discussed here also. So, prophase 1 includes leptothene, zygotene, pachythene, diplothene, and diakinesis. Now, I would like to ask you a question. You might be very much familiar with Hindi, और हिंदी लैंग्वेज में हम सब स्पीक करते हैं. So I basically try to make mnemonics so that it is very easy for the students to remember. आप इन पांचों सब स्टेजेस को याद कैसे रखें? It's very easy to remember them. इनको रिमेंबर करने का एक फॉर्मूला है, that is जब आपको प्यास लगती है तो आपको पानी की जरूरत होती है. So you just say लता जरा पानी दे. So L for leptotine, Z for zygotine, P for paclitine, D for diplotine, and lastly for dikinesis. So here you have to remember that the last D stands for dikinesis, and the mnemonics is going to help you a lot, and you have to remember the first alphabet. The first alphabet is important because L for leptotine, Z for zygotine, P for paclitine, D for first D, that is diplotine, and last D is for dikinesis. So I hope you got it. Is that clear? So it's very, very easy to remember these substages also, and the question comes that when does this crossing over start? This crossing over starts in paclitine. Now what is this? Let me tell you this. I'll show you how this crossing over takes place and how the exchange of genetic material takes place. Let's see that. Let's talk about this. You can see this. Suppose this is one chromosome. Okay. And this is another chromosome. So, here what happens is that, suppose I label it like this, because these are different chromosomes. ये अलग-अलग chromosomes हैं, तो इनकी labeling हम अलग-अलग करेंगे बच्चों. So here you can see this. This is a separate chromosome and this is a separate chromosome. So here you can easily see that this point where the genetic exchange is going to take place is called as chiasma or chiasma. This is called as chiasma or chiasmator. At this particular point, at these points, the genetic exchange will take place, and that's why we are different from each other. That's why we are different from our parents. That's why we are different from our siblings. Let's see how. So as a result of this, when the genetic exchange takes place, these are basically non-sister chromatids of homologous chromosomes. So how it takes place, you will see that. Let's see. Here you will be having like this. This part of this particular chromosome has come, and here also like this. Are you able to see this? This part of the second chromosome has come into the first chromosome. Similarly, in the second chromosome, what happens, you can easily see this part of the first chromosome has come into the second chromosome. This is crossover. यही है क्रॉसिंग ओवर जिसकी वजह से हम अपने पेरेंट्स से पूरी तरह 100% सिमिलर नहीं हो सकते, और यही है वो क्रॉसिंग ओवर जिसकी वजह से ब्रदर और सिस्टर भी, या दो बहनें या दो भाई भी आपस में 100% सिमिलर नहीं हो सकते. So the study of heredity and variations is called as genetics. तो जेनेटिक्स की जब हम बात करते हैं तो आपको पता है कि your father is the person who is responsible for running the household. उसी तरह, क्योंकि एक मेल डोमिनेटेड सोसाइटी रही है, उसी तरह साइंस में भी the person who has been responsible for establishing a particular branch of science or biology has been called as the father of that particular branch. For example, ये genetics जो है, इसका father हम कहते हैं Gregor Johann Mendel. So write it down. Father of genetics is Gregor Johann Mendel, G.J. Mendel. I hope it is clear. So when we talk about the father of genetics, Gregor Johann Mendel was a person who did so many experiments in the field of genetics that whatever genetics we are able to see today, that is because of the efforts of this man. But the story doesn't end here. Initially, when he did his experiments were not accepted. He published his work in Proceedings of Natural Society. But initially, people did not understand his work and people rejected his work. Initially, his work was totally rejected. And you know what he did? He was an Austrian monk. He was from a very poor family. He was working in a monastery. But he was having a lot of interest in natural science as well as botany, as well as in different aspects of plants. So, इसके दिमाग में हमेशा आता था कि प्लांट्स में क्यों होता है, कैसे ये अलग अलग तरह की वैरायटीज हैं, कैसे ये वैरायटीज अलग अलग तरह से प्रोलिफरेट कर रही हैं. तो he was having some knowledge of mathematics also. He was having a good knowledge of literature, good knowledge of philosophy, mathematics as well as biology. तो इसके दिमाग में ये आया कि हम क्यों ना ऐसा करें कि pure line varieties बनाएं. So what he did was he prepared some pure line varieties, but he worked on a plant which is called as Pisum setae. This is garden. This is garden. Now, जो आप garden में आपने जो मटर आप देखते हो वो यही Pisum setae. And you know the name of the genus is written like this, in which the first alphabet is always written in capital letters, whereas the name of the species is written like this, in which all the alphabets are written in small. It needs to be underlined as per the rules of the binomial nomenclature. You know that. And Pisum setae में he studied what? Different characters. Now, let's see what are characters. Suppose seed color. Now, the color of the seed of Pisum setae was a character. It is a character which is being studied even today also. But in this character, there can be different versions. So, different versions of a character are called as traits. Different versions of a character are called as traits. So let's see. Write it down. What are traits? Traits are different versions of a character. Now, what are the different versions of seed color? It can be yellow, or it can be green. Are you able to get that? समझ में आ रहा है? बिल्कुल clear cut समझ में आना चाहिए बच्चों. Genetics is a very very interesting subject, provided you understand it thoroughly. तो दो ही traits थे इसके जो मेंडल ने स्टडी करे, और दो ही traits normally आपको दिखते भी हैं. One is yellow and the other is green. So if we talk about yellow, it is found to be dominant. So Mendel said that there can be two versions of a character, and one of them is dominant and the other is recessive. Mendel ने उस समय को gene word नहीं कहा था. He did not call it as gene. He did not call these versions as alleles. He simply called them as factors. He simply called them as factors. So ये जो word था factors, यही काफी दिनों तक चला. And Mendel studied 7 different contrasting characters. And 7 pairs of contrasting characters were studied by him. So basically, in every pair of contrasting characters, he was able to find out that one of them was found to be dominant over the other, and the other was found to be recessive. So let's see how he did that. We go for a cross as well. Now let's talk about the monohybrid cross. I'll tell you how it takes place. Basically, Mendel studied different characters, and when he studied them, one of them was the height, one of them was seed color, one of them was the flower position. So you can study all these things. There were 7 pairs of contrasting characters, and in case of height, he took one of them as dominant. I'll give you a few examples. Dominant and recessive. So let's see that. In case of height, the dominant one was tall and the recessive one was dwarf. In case of seed color, Yellow was dominant and green was recessive. Similarly, in case of flower position, axial was dominant and terminal was recessive. Like this, it took 7 different pairs of characters into consideration. We are not going to talk about them. It is already mentioned in your textbook as well, and we will discuss about it in detail in the next class. But here, what I would like to tell you is that you can take any of these characters, just like Mendel took all of them. So, agar hum suppose height lete hain, what Mendel did was, he said that the generation which is taken initially is called as the parental generation. He denoted it by capital P, and he said that in a person or in an individual, in a plant, in an organism, in an animal, any organism for that matter, one allele comes from the male, that is the father, and the other allele comes from the female parent. But here, he took one of them as capital T, that is, he called them as homozygous. Why it is homozygous? Homo means same, and zygous means the individual having the race. So it means this particular individual, this particular organism is having same kind of alleles, that is capital T and capital T. That is the gene or the allele for tolerance. The other one was represented as small t. So the dominant one was taken as capital T, and the other one was taken as small t. So these were crossed, and this was taken as homozygous again. इसमें भी दोनों small t है यानी same है, but ये homozygous dwarf है बच्चों, and the other one is homozygous dwarf. Is that clear? Is it fine? Now, then he studied what are the gametes which are formed. So you can see the gametes. It will be capital T again. Here it will be what? Small t and small t. Can you see that? I'm encircling it to make it easy for you. Is that clear? Is it fine? Now comes the first filial generation. The generation which comes after the parental generation is called as the first filial generation. Suppose आपके मम्मी पापा के generation को अगर हम parental generation मान लें, तो आपकी generation first filial generation कहलाएगी, जिसको हम denote करते हैं by F1. So, what are the probabilities here? You can see this. Just cross it like this. It will be like this. Capital T, small t. It will be a heterozygous one. Can you see that? Yes. Capital T, small t. Again, here this combination will be formed, and here this combination will be formed. Is that clear? I hope it is fine. So, next one is what? This was taken for selfie. What is selfie? Do you know what is selfie? When you use your mobile and take a selfie of yourself, that is called as selfie. खुद की जब आप फोटो निकालते हो उसको कहते हैं सेल्फी. तो सेल्फी का मतलब क्या हुआ? To cross this particular variety with the same kind of variety, that is called as selfie. Is that clear? I hope it is clear. See, in biology you need to make the concepts familiar by relating it with day-to-day life examples. So here the selfie was done, and what happened was, you can see this, a kind of square was made by Mendel in his cross, but this square was proposed by some other person. So this was denoted like male. This is the symbol for male, and this is the symbol for female. So here you can see this will be capital T, small t. Why? See, these are heterozygous individuals. So basically we call these as heterozygous. But again, don't. Why? Because according to Mendel, if there are two alleles present, in those cases where two alleles are present together, only the dominant allele will be expressed if it is present along with the recessive allele. And the recessive allele will be expressed only when it is present in homozygous condition like in this condition. So, selfing of F1 generation or F1 hybrid will result in the next generation, which is called as F2 generation, which is also called as second filial generation. Second filial generation. Is that clear? So here you can see this is capital T, this is small t, and here you can see this will be homozygous tall, T and T. This will be what? Heterozygous tall, this will be again heterozygous tall, and this will be homozygous tall. So this is called as Punnett square. Punnett square. This is Punnett square. So the ratio was calculated in the F2 generation. So what was the ratio which came out? There were two ratios which were calculated. I will write it here. One, before going for those ratios, let's see what are these terms. One term is called as phenotype, and the other is called as genotype. I would like to tell you that this lecture we will be ending in half an hour. हम आधे घंटे में इस लेक्चर को खत्म करेंगे, लेकिन इसके आगे के टॉपिक्स हम आगे के लेक्चर्स में कंटिन्यू करते जाएंगे. So this is going to be really very interesting. तो जैसे ही आधे घंटे का ये लेक्चर खत्म होगा, उसके बाद I'll tell you how to go for the further lectures. तो चलिए जल्दी से हम देखेंगे. आप बच्चे मुझे बता दीजिएगा कि आधे घंटे कब खत्म होगा, ठीक है? चलिए, because I have to go for some other lectures as well. तो यहाँ पे हम देखेंगे phenotype में क्या है. In phenotype, what happens is that phenotype means the external characteristics. External characteristics means what? The morphological characteristics. Morphological characteristics. So these are the characteristics which can be observed externally. Which can be observed externally. बाहर से दिखने में कैसा है? Whether it is a tall individual, whether it is a dwarf individual, मोटा है, पतला है, छोटा है, जो भी है, that is phenotype. Genotype is the genetic constitution of an organism. किसी भी organism का जो genetic constitution है, that is called as genotype. So when we talk about phenotype and genotype, I hope the difference is clear to you. In short, we can say that phenotype is the expression of the genotype. Phenotype is the expression of the genotype. So, here you have to understand this, that based upon this definition, it came to Mendel's mind. Yeah, मैं कोशिश करता हूँ कि let me try to find out the genotypic and the phenotypic ratios. So let's see what we got. उस समय तक allele term was not coined. Even gene term was coined later on. Mendel had called these as factors, dominant factors and recessive factors. So देखिए ध्यान से क्या है. We were talking about this F2 generation. तो इस F2 generation में, here I will write to you the things. Phenotypic ratio क्या है? The phenotypic ratio will be what? Can you see this? This will be tall. This will be again tall, and this will be again tall. But this will be put across here. So this will be what? Dwarf. So it will be what? 3 tall is to 1 dwarf. Is it clear? 3 tall is to 1 dwarf. So the phenotypic ratio came out to be 3 is to 1. Phenotypic ratio came out to be 3 is to 1. And the genotypic ratio was how much? Let's see. See, this capital T is homozygous tall. These two, this one, I'm writing alpha here and alpha here, so that you can recognize them easily. These two are heterozygous tall, and this third one is homozygous tall. So it will be 1 homozygous tall, that is 1 capital T is to 2 heterozygous tall, that is capital T small t is to 1 small t small t. Is that clear? So this will be what? This is 1 is to 2 is to 4. This is the genotypic ratio in case of this cross, and this cross is called as monohybrid cross. Why do you call it as monohybrid cross, or why monohybrid cross name or term was given by Mendel to this cross? Mono means one, and hybrid means what? one pair of traits. One pair of traits or one character has been taken, and that character is what in this case? That is the stem plant. And the pair of traits, that is the two versions of that character, are tallness and dwarfness. Is that clear? So this brings us to a very important thing, that this is the phenotypic ratio, that is 3:1, and the genotypic ratio is 1:2:1 in case of Mendelian monohybrid cross. Now, if in a competitive exam or even in your university exams, they might ask you a question. They might give you a multiple choice question, or they might give you an objective question, that which one of the following is the Mendelian monohybrid ratio? They give you the first choice as 3:1, the second choice as 1:2:1, third choice as 2:1, and fourth choice as none of these. What would be your answer? क्या होगा आपका आंसर? अब तो ये दोनों दिए हुए चॉइस, और ऐसा भी नहीं कि both of these दिया है. अगर both of these दिया है, both of these is given, the answer would be both of these. मतलब अगर दोनों में से कोई एक चुनना हो तो किसको चुनेंगे? Any answer? Okay, let me tell you. The answer will be 3:1, because phenotype is the expression of the gene. And ये जो 3:1 है, ये आपको दिखता है बस. 1:2:1 का रेशियो पता लगाना पड़ता है. This you are not able to observe from outside, but this is a fact that phenotype is just the expression of the genotype. Is that clear? Okay. So this is all about the Mendelian monohybrid cross, and this cross was the first cross done by Mendel. So what Mendel did was that he proposed several laws, and one of them was the law of dominance. Now the law of dominance is not considered as a fundamental law by many scientists. Now the question is, why it is not considered a fundamental law? See, fundamental law is something which is not having any exceptions. Fundamental law is something which is not having any exceptions. For example, Newton's third law of motion. It says that every action has equal and opposite reaction. तुम दीवार को पुश करोगे तो दीवार भी एक फोर्स लगाती है. अगर आप किसी को कुछ कहते हो तो अगला भी रिएक्ट करता है. ये तो डे टू डे लाइफ एग्जांपल दिए मैंने आपको, लेकिन कोई भी ऐसा लॉ जिसका कोई एक्सेप्शन ना हो, दैट इज कॉल्ड एज अ फंडामेंटल लॉ. बट मेंडल ने 3 लॉज दिए थे. उनमें एक था लॉ ऑफ डोमिनेंस, एक था लॉ ऑफ segregation, and the third one was law of independent assortment. So here I would like to tell you that ये जो law of dominance है, this is not considered as a fundamental law by many scientists, and that's why मेंडल के 2 ही लॉज माने जाते हैं, लॉ ऑफ सेग्रीगेशन एंड लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट. मेंडल का ये लॉ ऑफ डोमिनेंस फंडामेंटल लॉ नहीं माना जाता. देयर आर मेनी एक्सेप्शंस टू दिस, एंड एक्सेप्शंस इंक्लूड इनकम्प्लीट डोमिनेंस. द एक्सेप्शंस इंक्लूड को-डोमिनेंस. इनकम्प्लीट डोमिनेंस मतलब 2 एलील्स हैं, जैसे मेरा एलील चलाता हूं. देयर आर 2 एलील्स. फॉर capital R is representing for red flower varieties and small r is there for white flower varieties. But when you cross capital RR into small r, that is homozygous, red flower variety with homozygous white flower variety, you are getting pink flower variety. So that is incomplete dominance. Co-dominance, for example, in our blood group system, when I stands for immunoglobulin. So when IA and IB are present together, that gives rise to not A or B blood group, but AB blood group. So this is an example of what? Co-dominance. Similarly, in blood group system, there are 3 alleles. One is IA. I stands for immunoglobulin. The other allele is IB, and the third allele is IO, which is also called as small I. So both of these are co-dominant, and the third allele is also present. So there are more than 2 alleles for a particular character, जो कि मेंडल के लॉ ऑफ डोमिनेंस के हिसाब से पॉसिबल नहीं था. मेंडल के लॉ ऑफ डोमिनेंस के हिसाब से सिर्फ 2 ही alleles पॉसिबल थी, in which one allele is dominant and the other is recessive. So these exceptions have resulted in a particular thing. And what is that particular thing? That this law, that is law of dominance, which was given by Mendel, is not considered a fundamental law, and hence we consider only 2 fundamental laws of Mendel, that is law of segregation and law of independent assortment. In the next class, I will start with law of dominance. I will tell about this particular law and why it is not considered a fundamental law, and then I will tell you about the exceptions like incomplete dominance, codominance, and multiple allelism in detail, and then we will go into the further details of law of segregation and law of independent assortment, which are considered as the fundamental laws of genetics. So I will also tell you about the reasons for the success of Mendel and why Mendel's laws were initially rejected and who were the rediscoverers of Mendel's laws, how the genetics has reached up to this height. We will discuss these in the further classes. Thank you.